Hi, this is our GMO project on anti-inflammatory genes in the sweet orange citrus senescens. I'm Melanie LeMinh.
I'm Ashley Darst.
And I'm Mackenzie Molina.
The organism that we chose to modify was a citrus senescens, and it contains anti-inflammatory properties due to flavonoids. These flavonoids are natural phytonutrients that can help in heart health, as well as reducing the risk of cancers like breast and ovarian cancers. The main purpose of flavonoids, however, is to reduce inflammation in the body, and they can reduce the risk of chronic inflammatory diseases such as arthritis, metabolic inflammation, and age-related inflammation, which affects 60% of Americans. The reason we chose citrus senescens as our organism is because of its anti-inflammatory properties, and it would be a natural alternative to anti-inflammatory medications, which have led to stomach ulcers and GI tract bleeding when used in large doses. Chronic inflammatory diseases affect many Americans, and a genetically modified orange with an increase in flavonoids could be a good natural prevention method for many of these diseases.
Biosynthesis pathway for flavonoids. As you can see in the right-hand flowchart for the biosynthesis pathway, we have boxed the CHS genes in orange. These genes encode enzymes that help regulate the biosynthesis of flavonols, which are just a type of flavonoid. In citrus sinensis, there are two types of this gene. There's the CHS1 and the CHS2 genes. In this project, we will be focusing on the CHS1 gene. To increase flavonoid production in citrus sinensis, we will genetically modify the plant to increase the expression of the CHS1 gene with a promoter associated with higher rates of expression. We chose to use the cauliflower mosaic virus 35S promoter, which is commonly used in plants. This promoter is found in the plasmid we are using for our genetic modification. This plasmid will be inserted into our plant using agrobacterium.
This modification is usable and innovative because the CHS gene is already encoded in the citrus sinensis genome, and this modification of flavonoids has not yet been attempted. However, a similar experiment involving the citrus jamboree, also known as the rough lemon, was successfully modified by using a similar procedure of modification to the one we are proposing. By using agrobacterium tumefaciens and a plasmid, the Hal2 gene was inserted into the lemon's genome to increase salt tolerance for growth in harsh conditions. In addition, because the CHS1 gene is from the citrus sinensis organism itself, there will be a greater chance for successful integration in the flavonoid biosynthesis pathway, thereby increasing feasibility. Finally, this modification is innovative because we are using the organism's own properties to our advantage.
For this project, we would produce 50 genetically modified citrus sinensis plants, and we would use RNA from an unmodified citrus sinensis to perform this modification. We would insert the gene of interest, the CHS1 gene, into a binary vector, which would then be inserted into the Agrobacterium tumefaciens so that it could be transferred into the plant's genome. The binary vector that we chose for this project contained the cauliflower mosaic virus 35S promoter that is known to produce high rates of expression in the genes in the plants that they are typically associated in. The cauliflower mosaic virus promoter will be used to increase the expression of the CHS1 gene, which will then increase the synthesis of flavonoids in the modified orange.
First in this process, RNA will be isolated from the citrus sinensis plant since it lacks introns. RT-PCR will be used for this RNA into single-stranded DNA, which can then be used for normal PCR. This PCR will be used with primers that contain linkers to the restriction enzyme sites shown to the right. These sites are BSIWI and BSRGI, which are found in the binary vector that we will be using. The primers contain linkers that correspond to these restriction enzyme sites. The CHS1 gene will then be inserted into the P green 2 binary vector between the left and right tDNA borders in between these restriction sites. DNA ligase will be used to bind the DNA strands together to create one continuous vector. The P green 2 vector is inserted into Agrobacterium tumefaciens using electroporation. In this experiment, we are using the LBA 4404 Agrobacterium cells, which include the helper plasmid PAL 4404, which contains the VIR genes to aid in the transfer of the tDNA from the vector into the plant cell. Next, Agrobacterium cells are plated on agar plates, and white-blue selection is used to determine if the vectors were taken up by the cells. This can be done since the vectors contain the LAGZ gene. A scalpel will be used to pick up an Agrobacterium colony that successfully took up the vector. A leaf from Citrus sinensis is cut with a scalpel with the colony so the colony can be inserted into the plant's wound.
The agrobacterium attaches to the plant's cell wall and forms a connection between the two via a T pilus created by the VIR proteins. The gene insert in the tDNA region of the P green 2 plasmid is transferred into the plant cell. The VIR proteins from the helper plasmid then chaperone the tDNA, which is inserted into the plant genome. The treated leaf is cultured on an agar plate but formed calluses. These new calluses are the part of the plant that contain the tDNA coding for the higher expression of CHS1 and hopefully have an increased flavonoid production. The calluses are removed and further cultured on agar plates containing hormones needed for plant growth and kanamycin to select for transformed cells. Kanamycin resistance is a selectable marker in the tDNA, which would have been transferred to the calluses on the plants. This antibiotic is used to test the cells for successful expression of the tDNA in the transformed cells. Once the calluses have rooted, they are transferred to pots and cared for until they grow into full orange trees.
The helper plasmid, shown on the left, comes with the agrobacterium that we would purchase for this project. On the helper plasmid, there are the VIR genes that help the agrobacterium transfer tDNA to the host organism. The binary vector on the right contains the tDNA region that the gene of interest, the CHS1 gene, is inserted into between the two restriction sites BSIWI and BSRGI. This pea green vector has a LAGZ gene to be used for blue-white selection, a cauliflower mosaic promoter to increase the expression of the gene of interest, and the CAN-R selective marker to test for antibiotic resistance and the uptake of the plasmid into the agrobacterium. Together, these two plasmids work together in a binary system.
In order to test if the uptake of the tDNA actually increases flavonoid content in the orange, we are going to use liquid chromatography and HPLC. In this process, a water solution will be set to a speed of 1 milliliter per minute and injected into the orange in a volume of 10 microliters. The orange juice will be squeezed out and then centrifuged at a speed of 3,000 RPMs for 10 minutes. The pH of the juice will then be adjusted using sodium hydroxide until the pH reaches 8.5. This juice is then filtered through a special membrane of 0.45 micrometers and analyzed using HPLC, which contains a pump, a UV spectrophotometer, and software for analysis. The spikes in the resultant graph from this data will then show us the flavonoid content, specifically hesperidine.
In order to quantify statistical success, a t-test is conducted in order to test the null hypothesis. If the p-value obtained from the results is less than or equal to 0.05, the null hypothesis can be rejected because it means that significant results have been obtained. The hopeful result is that there is a significant increase in flavonoids.
In the diagram, it shows a phylogenetic tree for the CHS1 gene found in citrus sinensis. Based on the tree, the gene is an ortholog and is fairly common among many citrus fruits that could code for the synthesis of flavonoids. Due to the gene being fairly common in other fruits, if the modification of citrus sinensis does not work, other fruits could be modified instead. A possible consequence of introducing the modified citrus sinensis into the environment is a chance of polyploidy and breeding with other orange species, which could lead to some forms of hybrid breakdown.
In all GMOs, there's a potential for long-term effects. This includes increase in antibiotic resistance, allergies triggered by consumption, and exposure to toxic proteins. For our case, excessive flavonoid intake has been found to act as a mutagen in high doses above 68 milligrams to be specific. These adverse effects of flavonoids outweigh their beneficial ones. For this project, we would need to test to see if the flavonoid content in our modified oranges is above or at 68 milligrams. And if so, we would need to change our proposal in order to reduce the flavonoid content. Additionally, GMO technology can be exploited by large corporations and governments, like in the case of the Coca-Cola controversy in 2014, where African migrant workers were exploited by the Italian government through orange plantations. This case study is pertinent to our proposal since we are working with the orange species Citrus sinensis. We will need to work with local governments and international organizations to assure ethical economics and working conditions when our orange species is used.
Neomycin, kanamycin, and geneticin are antibiotics within the tDNA of the plasmid being utilized to genetically modify Citrus sinensis. If Citrus sinensis breeds with local plants, the antibiotic resistance may be conferred to the wild population. Conference could affect crop yields and availability of food. To combat this, these modified organisms would be grown in an isolated area. In addition, the proposed genetically modified Citrus sinensis would need to be approved by the Institutional Review Board for clinical trials to be performed on human subjects. Human trials would be pursued in order to receive licensing as an over-the-counter drug. Reviews of the drug should be conducted often to assess exposure risks to the environment, and risks or complications due to human or animal consumption.
For our future directions, the modified Citrus sinensis could supplement or replace current non-steroidal anti-inflammatory drugs such as Advil, ibuprofen, and Flanax, as more research on their safety and efficacy is performed by agencies such as the FDA. Other citrus fruits, such as Citrus subicima and Citrus clementina, could be modified in a similar fashion to increase their synthesis of flavonoids. Another direction could be to not only enhance the anti-inflammatory properties of the Citrus sinensis, but increase the vitamin and healthy fat contents in the orange as well. This genetic modification could be done in a similar manner as the anti-inflammatory modification by using a separate promoter and gene specific to the vitamins and fatty acids via an agrobacterium vector.
If our proposal were to fail, meaning that the CHSI genes more expressed in the Citrus sinensis did not produce a higher content of flavonoids, there are two other genes we can use to achieve this goal. These are the other CHS gene, the CHS2 gene, and the CHI gene, which encodes for chalcone flavonoid isomerase, and is one step lower in the biosynthesis process for flavonoids. We could either insert these genes separately with a cauliflower mosaic virus promoter, as we did with the CHS1 gene, or we can insert all three in combination in order to try to increase the flavonoid content in Citrus sinensis.
Thank you for listening to our GMO presentation on how to modify citrus sinensis to increase flavonoid production in order to help inflammatory diseases.